should finish up with the male reproductive system. We just sort of started last time. So again, primary reproductive organ of the testes that produces the male gamete, the sperm. And I think where we sort of, a little bit we did last time, the testes, they originate basically for tissue that's right next to the ovaries. They're kind of in the same spot in the early embryo. But then the males in late development, there's a little contractile bit, the gubernaculum that is attached to the testes and kind of the floor of what's going to be the scrotum. It contracts and draws the testes out there. Why are the testes in that scrotum? It's kind of a stupid place to keep them, in some perspectives. But that's to keep them cooler because mammals can't produce viable sperm at body temperature. So they got to be out there to cool a little bit. And then we wrapped up, I kind of went off on a tangent on the serous membranes. In the abdominal cavity, the two serous membranes are the peritoneum. Again, parietal covers the lines, the cavity. Visceral then folds back and covers each individual organ. So most of your abdominal organs, the outer layer is the visceral peritoneum. Now, it may be called different things, like in the uterus, this diagram, there is a visceral peritoneum. You can call it the visceral peritoneum. It's the outer layer of the uterus and the inside. It's also called perimeter. We look at a lot, but it's the same thing. You call it perimeter or visceral peritoneum. You're fine. Usually, when we're referring to the organ, they use perimeter if they're referring to the serous membranes, no visceral peritoneum. But it is the same structure. Either name you're talking about the same thing, except either one. But you'll find that with most organs internally, they're going to have a serous membrane around the outside, with a couple exceptions. So the testes drag themselves down. As they come down, of course, it's not just the testicle rattling around in the scrotum. It drags down a lot of other stuff with it. Of course, the testes have to have blood vessels, nerves, ducts to conduct the sperm. You drag down a piece of that parietal peritoneum, which ends up down in the scrotum. So let's look at a couple other things that play a role in controlling the temperature of the testes. Let me turn off one of these lights so you can see. All right, so we're looking at front view here. They've removed the scrotum, which is just integument, that's just skin. So you're seeing, this is the inguinal canal. So up here would be the abdominal cavity. So the testes ascend down through the floor of the abdominal cavity into the scrotum. Of course, that is the inguinal canal. That's the opening it passed through. And there's still stuff there, of course. All of this whole tube that's drawn down from the testes, that is, from lab, the spermatic cord. It's got a connective tissue covering. You're showing it partly reflected here. And within that connective tissue covering, there's several things. So let's look at a couple on your left here. All right, as the testes descend, here's your spermatic cord. Notice inside that connective tissue sheath, there is some muscle. It's actually skeletal muscle that is drawn down with the testes through the spermatic cord and into the scrotum. That is the cremaster muscle. So what it is, you remember last semester maybe you dissected the cat, you had the transverse abdominal, internal, external obliques, right? Because those are abdominal muscles. Basically, the testes are pulling some of those down with them. I mean, this, if you follow it up, it started up here. There's your internal obliques right there. So some abdominal skeletal muscles are pulled down. So in addition to the cremaster muscle, which is a skeletal muscle that just gets pulled down with the testes into the scrotum, if you look at the scrotum itself, it is just, of course, skin and integument, but there is some smooth muscle in the walls of the scrotum. It's called the dartos muscle. Now, why do you have muscles associated with your scrotum and your testes? I like looking at it, Brandon. It's temperature control. So part of the reason you have these muscles down there, if you imagine, here's your abdominal cavity. That's the heat source. That's the core, your core temperature, abdomen. Then your testicles down here somewhere. Well, if your testes get too warm because they're pulled up against the floor of your abdominal cavity, they'll get too hot. So what you can do is relax the cremaster and dartos muscle. That allows the testes to drop further away from the heat source. They'll get cooler. On the other hand, you don't want your testes too cold because then they can't produce sperm effectively either. So if they get too cold, you can contract the cremaster and dartos muscle. Cremaster, of course, will just shorten up the spermatic cord, which will pull the testes up. The dartos muscles in the wall of the scrotum, when they're relaxed, the wall of the scrotum is all flaccid, kind of hangs down lower. when the dark muscle contracts, it kind of makes the scrotum all wrinkly and sucks the testes up closer to the floor of the abdomen, which is the heat source, so they don't get too cold. So generally, premascular dark muscles are used to control the distance of the testes away from the abdominal floor. That's to help control the temperature. That's their main role. They do have another role. It's like that Michelangelo made that statue of David, and it's somewhat lifelike. It's pretty accurate. But people point out his genitalia are kind of small. All right, I think he was about to, they were about to bash each other with swords or something like that. So if you're going to be bashing someone with swords, you don't want your scrotum and testes hanging out there because they can get damaged. So part of the fight or flight response will also affect these muscles. They contract and suck everything in really close so they don't get smashed or chopped with a sword. So they will be controlled by the fight or flight response as well. But ordinarily, it's just temperature control. So developmentally, we route the testes out of the scrotum to keep it cooler. Then we have these muscles that can change their distance away from the heat source. Again, temperature regulation. And finally, there's one other problem. In lab, in your lab sheet, you have testicular arteries and veins. Of course, testes have metabolism. They need raw materials. They need oxygen. So they have a blood supply. Arteries bring oxygenated blood to the organs. They're typically shaded in red. That's just the color they use for oxygenated blood. In this case, it is the testicular arteries. And of course, the blood passes through the testes, then it returns to the circulation in the testicular veins, which are shaded in blue on this diagram and our models. Here's a potential problem though. You got your abdomen up here, really high temperature. You're going to take high temperature blood, send it down to your testes down here, and then of course that blood will go back to the circulation. How are you going to keep your testes cool if you keep pouring 98.6 degree blood on them? It's this constant heat source. You can't get rid of it. There is a mechanism associated with testicular arteries and veins that helps to reduce the heat load on the testes. It's something you see commonly in physiology. It's called a countercurrent heat exchanger. Lots of animals use it with their circulatory system if you need to control the temperature in various organs. Sharks use it to keep their brain warm, some fish use it to keep some muscles warm, so it's fairly common. But all it is, it's called a countercurrent exchange mechanism. Countercurrent just means the fluids are moving in opposite directions. Very simplified view here, what you're seeing, this is an artery, so it's bringing warm blood, 37 Celsius is body temperature, not Fahrenheit on this diagram. But that blood is very warm, you can't just pour it directly out the testes, it'd overheat them. So what happens, the vein, in this diagram is not quite accurate. The vein and the artery, they're smushed right next to each other. They are in mechanical contact. This allows conduction of heat. Heat moves a little bit like diffusion. Heat will always move from an area of high heat to an area of lower heat. So if you physically sort of connect these two blood vessels next to each other, when this hot blood comes down here, this blood returning to the testes has been cooled. It radiated some heat out in the environment. So you have cool temperature blood here, in contact with the hot blood over here, and if those blood vessels are in contact, the heat will move in the direction of these little arrows, this direction. And because we're moving opposite direction, the blood in the veins is always cooler than the adjacent blood in the arteries, so heat will continue to, to move across. And then by the time this blood reaches the testes, some of the heat has been removed. Notice it starts at 37, ends at 35. In a similar manner, this cool blood coming back to the testes, you're actually warming it up before it gets back to the body. So it's a way to kind of take the heat and move it from the arterial blood over the venous blood so you don't overheat the testes. And it's pretty common. You ever wonder, you see those little shorebirds like running through like little dippers and things? They're standing in 33 degree water. They're just like us, they have a body temperature. If we stuck our feet in ice water, you'd be losing heat tremendously. They have the same thing in their legs, stuff like that. So it's a common mechanism to prevent heat loss or heat gain. It's also why, this is not what the testicular artery and vein looks like. A little more realistic is right here. You only need one artery to take blood to the testes, and you can have one or two veins bring it back. But look at this vein. That's not a vein, it's a whole network. It's like all these interconnected veins. Again, it's surface area. If you had two big blood vessels, there wouldn't be a large point of contact, there would be a lot of heat exchange. So instead, the arteries and veins branch into this mass of smaller blood vessels, each vein in contact with an artery, and that just gives you a lot more surface area for the movement of the heat. We're calling this peniform plexus. That's just that huge network of testicular veins. They branch a lot. It'll come up later. This is also a site of varicose veins in male. It's kind of a weak point in our circulatory system. Sometimes those valves will fail in those veins. They get all distended. All right, so we kind of got three things. Help control the temperature, keep the testes cool. One is developmental, put the testes down the scrotum. Two, dartos and cremaster muscle. And three, the countercurrent exchange for the circulatory system, for the blood. Those all help to keep the testicles cool. So let's go inward a little. We've got Wilson with his schematic for it. I want to name a couple other structures that are associated with the testes down in the scrotum. Of course, the scrotum's first. Imagine we remove that. Once you remove the scrotum, let me go to a simpler diagram here. So once you remove the scrotum, we'll do something like this. There's a little loosely attached membrane just partly covering the testes. This is what I showed you in lab in that goat testicle. It's not on your lab sheet, but this is that part of the primal peritoneum that gets pulled down with the testes. Once it's down here, it's called the tunica vaginalis. So that's just a loose fitting sort of partial bag around the testes. Then you cut it, the testes falls right out. They're not really connected. So if we get rid of that one, then the next layer, and this was one of your lab terms, there is a dense connective tissue sheet physically attached to the outside of the testes. This is what was called the tunica albuginea on your lab sheet. So this is attached to the testes. The tunica vaginalis will flop right off, but the tunica albuginea is physically attached to it. It's got the outer layer of the testes. And of course, it's mostly protected. Again, somewhat vital organs, you don't want them damaged. So that dense fibrous connective tissue protected it. It's a little bit, if you looked at like fascia on the muscles last semester, that's what the tunica albuginea is like. It's the dense connective tissue wrapping around the testes. Now, one thing to mention about it, the tunica albuginea forms all these little septal projections or plates that radiate into the inside of the testes. I can draw one with them. So inside the testes, we look at the microscope slide, what you have is a bunch of coiled up tubes, these yellow things here. These are the seminiferous tubules. So that's what we looked at in the slides. These are what make a sperm. There's tightly coiled up mass of seminiferous tubules. That's the main component in the testes. But this tunica albuginea makes all these little septa, or it grows in to the testes, separating into subcompartments or lobules. I just want to mention this because you'll see this in a lot of organs. A lot of your internal organs, pancreas, liver, lungs, many others, the organ is compartmentalized by connective tissue sheets. So these are sheets of connective tissue that separate the inside of the testes into smaller, basically subcompartments. It's the same idea as, putting pressurized, bulkheads in ships and things. These organs are essential. So if you get, a parasite or an infection or damage in one area, you don't want it to easily spread throughout the whole organ. Then it's toast, doesn't do anything, and you're not going to reproduce. Connective tissue is a pretty good barrier. Microbes don't get through it. It's relatively inert. Remember, it's just a bunch of sheets of protein for the most part, the matrix. So you'll find a lot of our organs that are compartmentalized like this. That way, if you get some damage or a parasite or something in here, it can't easily get spread to the rest of the organ and damage it. So you see that a lot. If you remember, when you looked at the pancreas, the endocrine system, did you notice it almost looked like it was chopped into bits? You had these white lines running through it. That's it. The pancreas is separated by connective tissue sheets as well. The lung is, a lot of organs are. It's just a way to compartmentalize damage or infections or parasites. So the tunica albuginea produces these little septal extensions that separate the testes into these little subcompartments or lobules. Within each subcompartment or each lobule, you'll have a handful of seminiferous tubules, 3 or 4 to 6, I think 3 to 6. Now what we're going to do at this point, I'm going to start at the seminiferous tubule, and we're simply going to follow the pathway of sperm from where it was produced until it leaves the male's reproductive system, so opportunity reproduction. It is one continuous tube, so the sperm have no option. When they're produced in the seminiferous tubule, the only way they can go is through all this long series of tubes and out. Now, some of these terms I'm going to go over now, these are the ones on your lab sheet with the asterisks. You can't, with a lot of training, you can find them on a section of the testes. I couldn't. I don't expect you to. Because they're all a bunch of tubes. When you take a cross section and make a slide, they kind of all look the same. So, I use a diagram like this where everything's laid out and color coded so you can kind of see the differences. So I'll go through them quickly. Like I said, they're on your lab sheet. I don't ask them in the lab. If I did have to get a diagram like this, you can't tell by looking at the sections of the testes under the microscope, not easily. So the seminiferous tubules, they'll produce the sperm. And then these seminiferous tubules, they're all connected to a little tube that leads out of the lobule. It's called the tubulus rectus. They Latinize all these things. Tubulus rectus, rectus means straight. It just means straight tube or straight duct. So we start at the seminiferous tubule. That's where the sperm will produce. As the sperm are produced, they'll make their way through these straight ducts. And you can call them straight ducts or tubulus rectus. I don't really care what, it's the same thing. Which will conduct the sperm into a different organ, a different structure, right along here. That's a network of interconnected tubes. So all the straight ducts go to this big network of tubes, which is called Reeves net. That's how we translate. Like even when you look at the adrenal cortex, have that zona glomerulosa, zona fasciculata, and zona reticularis. If something is reticulary, or it's a reed, that just means it looks like a net. It means net line. So it's just this net, sort of interconnecting tubes here that the sperm will pass through. And then the sperm will exit the testes and enter another organ through efferent ducts. Physiologically, afferent means going to, efferent means going away from. These efferent ducts are just taking the sperm away from the testes. That's kind of what the name means. And they deliver the sperm to the epididymis. So this thing here is a separate organ. It's not the testes. But it lays right alongside them in the scrotum. This is the epididymis. It is also tube-like, as all these things are at this point. They're roughly 20 feet long, I think, if you stretch it out. But the epididymis does a couple of things. Males hit sexual maturity, they're making something like 400 million sperm a day. It's a huge output of cells. But you might not need them right this minute, but you're going to continue to make them. So what happens is the sperm make their way into the epididymis for kind of short-term storage. They will sit there until ejaculation, where they'll leave. So kind of the function of the epididymis is short-term storage for the sperm as you're producing them. They'll just kind of pack away into the epididymis till needed. Also, the walls of the epididymis, they have microvilli, which indicates exchange of some material. So while the sperm are sitting there temporarily being stored in the epididymis, there's exchange of material. Some final maturation of the sperm does occur while it's sitting in the epididymis. I'm just looking for the diagrams, because it's all small bits. So up here, this one looks more or less like what we have in the lab. All right, so we're down here. We've taken the sperm from all the small ducts. You can't even see them in this diagram. Stored them in the epididymis. Now, when you need the sperm at ejaculation, the sperm are going to make their way from the epididymis into this tube here. Which is called the ductus deferens or the vas deferens. You can use either name. Vas deferens is a little older. I tend to use it, probably because I'm old, but ductus deferens means the same thing. That's what your textbook has in it. Now note on this diagram, this is like some of our models in the lab. There is no spermatic cord here. The ductus deferens is in the spermatic cord, but on a lot of these models, because they're trying to highlight just the reproductive ducts, they've stripped away most of the spermatic cord. There's no testicular artery and vein, there's no cremaster muscle, there's no connective tissue sheath. They just left the reproductive duct that wouldn't be moving through the spermatic cord. I don't really say that because in lab, you got two different things in the same spot. Some of the models are like this, everything's been stripped away. The only thing that's left is the vas deferens. In other models, they left the spermatic cord intact. Don't call it the vas deferens, because the vas deferens is in there, and you can't really see it though. There's a lot of other stuff in there, so the whole thing is called spermatic cord. So what the vas deferens is going to do, it's going to take the sperm, move it from the epididymos into the scrotum, and back up into the abdominal cavity. I want to mention a couple things about this though, because a couple weird things that happen here. There is a male surgical birth control technique where they'll go in and they will cut the vas deferens. That's analogous to a tubal ligation in females where they cut the uterine tubes. But in males, it's much easier because this isn't really even abdominal surgery. This thing's hanging out here. You know, I mean, shoot, I don't need training, but I can probably give someone a vasectomy. You just make a little incision, cut that tube, you're good to go. You know, you tie the ends up, cauterize. But you know, it's not that difficult. Of course, you might have that done. Let's say, oh, you got 6, 7 kids, you know, you're half broke. So, honey, I think we'll call it quits and just go and surgically snip your duct. Not as invasive as the tubal ligation. But what I was thinking is kind of funny. When you cut the vas deferens, of course, they got cut both, there is no sperm going to make it from the testes out anywhere else. So that individual should be functionally sterile. But all those surgical birth control techniques, they don't say it's 100% effective at preventing pregnancy, which seems not be 100% effective. Couple reasons why. If you go in to the scrotum, of course, there isn't a nice neat vas deferens hanging there. And it's a human doing a surgery. It might be the end of a 12-hour shift. I'll just cut that. We're good to go. Yep, that was a testicular vein. So there could be mistakes in surgery. They cut the wrong thing. What they'll normally do, they go in and they cut a section of the vas deferens out. They patch it up, send it to pathology. They make a slide of it. They look at it and go, yes, that is the vas deferens, not a vein, and you got the whole thing. So they usually check themselves. At least they're supposed to. But even if it's done properly, occasionally, there could still be residual sperm in the rest of the duct. So there's like a waiting period after your vasectomy. I think I'd be right up for it anyway. But you can have vasectomy say, wait so many days, and then the residual sperm should be neutralized and be good to go. But if you had a vasectomy, went home and had sex, there could be some sperm in there. They could still make their way out in there and have been some pregnancies for that one. But that's not the weird part. This is kind of weird. Let's say you do this. We go in, and you have your vasectomy. Everything's great. But then you win the lottery. Hey, you got $300 extra $1 million. Honey, let's have more kids. Let's make our own softball team. You know, it's great. So could you go in and have this surgery, reversed? And sure, I could probably do it. The thing's not that small. You go in there and cut the ends and sew them back together. The plumbing's reconnected, should be good to go. And usually it works. About 90% of the time, you reconnect the vas deferens, out come the sperm, more children. 10% of the time, it doesn't work. I think it's kind of weird. You reconnect all the plumbing. This tube is good. Everything's working. But 10% of the time, you reverse vasectomy. That male is still sterile. Doesn't have any children. Any idea why? Cool, why? All right. The reason why. Did you look at the blood-brain barrier last semester a little bit? Just mentioned it. All right, what that is, it's tight junctions in the blood vessels between basically the circulatory system and the brain, right? Gives you very tight control over what moves with circulation into the brain, because the brain's really sensitive. You get random chemicals from your blood getting in there, it kind of screws up its function, which is bad for us, kind of depending on our brain. Well, it turns out you also have a blood testicle barrier. Testes aren't that sensitive. Why blood testicle barrier? Same thing, tight junctions in the blood vessels. The reason why is your infection fight, your immune system. In simple terms, the way the immune system works, early development, your immune system sort of takes an inventory of all the proteins on your cells and in your blood. And the way it's supposed to work, if something comes into your body that's sort of not on the list, your immune system attacks it. That's how it works. Bacteria gets in there, it's got proteins that are not yours, you attack it. But the problem here is, when your immune system is maturing, what are your testes doing? When you're a year old, 6 months old, early late development. Testes aren't producing sperm, are they? You don't produce sperm, until sexual maturity. So all of a sudden, in puberty, you start producing these sperm. Sperm are genetically distinct from every other cell in your body. They express different genes. In fact, they're haploid. They don't have half the normal number of chromosomes, so they're completely different. So the potential problem is here, the sperm would be recognized as foreign invaders. Your immune system would attack them. So that blood-testes barrier is basically there to prevent your immune system from getting exposed to sperm antigens. And as long as it's intact, you're fine. You don't attack your own sperm, that's a good thing. But if you get a vasectomy, where you cut and tie out this thing, your testes are still making 400 million sperm a day. They got nowhere to go. They're just going to build up in here. And in some cases, because the sperm can't leave, that blood testicle barrier can get damaged. And more or less what you're doing is vaccinating yourself against your own sperm antigens. So you'll start making antibodies. Once your immune system is exposed to these new antigens, the sperm antigens or proteins, you'll start making antibodies which will go in and destroy the sperm. So even if you reconnect the vas deferens, your immune system's kind of destroying the sperm as fast as you're producing. Sperm counts stay real low. You can stay sterile. It doesn't happen much of the time. I think it's 5 or 10% of the time. But it can happen. It's kind of weird. Let's assume everything is still connected though. So we'll take the sperm now, and they are free to move through the vas deferens, back up through the inguinal canal, and into the abdominal cavity. Of course, how do the sperm move? We know sperm swim, right? They got that big long flagellum, that's what it's for, they swim. They don't swim in the male reproductive tract, it's too slow. In the male reproductive tract, sperm are moved by what we call peristaltic contractions. This small tube the vas deferens has smooth muscle in it. Many of your tubular organs work this way, your ureters, your intestines. But basically what happens, if sperm are in this tube, the smooth muscle in this area will constrict. When it constricts, what happens is sperm liquid there, they get squirted up forward. Then the next part of the muscle constricts. If you can watch this, you can see this ring of smooth muscle start at one end of the vas deferens, and that contraction move like a wave along the whole vas deferens, propelling the sperm in front of it. The analogy you can use, take a thin walled plastic tube filled with Play-Doh, put your fist around it, keep your fist clenched, and slide it down the tube. What's going to happen? You're going to push all the Play-Doh in front of it, right? That's basically what a peristaltic contraction is doing. That moves the sperm through the male reproductive tract. So the sperm aren't really swimming yet. The peristaltic contractions are what move them. And so they will be propelled in the abdominal cavity. This thing here is urinary bladder. Of course, you have two vas deferens, one coming from each side. They kind of loop up over the bladder and around it, and then come down towards the bottom of the pelvic cavity here. So just keep in mind, when we're looking at one half here, there's two vas deferens coming in. And they will move along, move along, move along, move along. Right here. That is the end of the vas deferens. Of course, the tube is going to continue until you leave the penile urethra, but we change its name at different points. So at this point, this is the end of what we call the ductus deferens. That's it. Now, what's going to happen at this point? There are a number of accessory reproductive glands. They contribute actually most of the semen volume. Sperm that are produced by the seminiferous tubules, that only accounts for about 5% of the ejaculate volume. So sperm is not a huge volume. That's why if you have a vasectomy, there's still ejaculate and it's still almost the same volume. You're only losing 5% of its volume because you don't have the sperm anymore. So these accessory reproductive glands, they include, there's 3 of them. Seminal vesicles, right here. Yeah, there's 2 of them, one kind of at the end of each vas deferens, and one on the other side of the board here. So there's the duct there. The duct from the seminal vesicle, which is a gland, come out here, and they join with the vas deferens. This gland, the seminal vesicles, produces actually most of the semen volume. Now, all you have to do with these accessory reproductive glands, kind of list their contribution to the semen. By that I mean the volume they add and the main components. Seminal vesicles, they're responsible for about 60% of the semen volume is from the seminal vesicles, the majority of it. And of course, these secretions are all aqueous solution. They're water, there's ions, electrolytes, and other things in it. So I won't sort of go through the whole list. Just keep that in mind. These are mixtures of lots of compounds. But one of the important components of the secretions from the seminal vesicle is they contain the carbohydrate fructose. It's carbon sugar, not like glucose. Why would you have sucrose in your ejaculate? It just seems kind of wasteful. Like glucose, again, fructose, glucose are more or less interchangeable. But fructose is a carbohydrate. It's an energy source. Probably its main function, if you think of the sperm, all right, at the anterior end of the sperm, what the lab we call the head of the sperm, there's a nucleus with the DNA. That's going to be the male's genetic contribution. There's a small membrane-bound organelle I'll talk about later, acrosome. Then you got the body. In this body, if you look inside it, it's not very large, but it contains, these are supposed to be mitochondria. And then you got the big long flagellum. All right, a couple things about sperm. These are the tiny cells you find in the body. They're really thin. You went over meiosis and mitosis last semester, at least a little bit, right? Right. Mitosis occurs in all your somatic cells, your regular body cells. The only place where meiosis occurs will be in your gametes, your reproductive cells. So if you're male, the only place you use meiosis or meiotic divisions is to produce sperm. The other place females use meiosis is to produce the eggs or oocyte. It doesn't happen anywhere else. So recall, sperm are a little different. I already mentioned they're haploid. They only have 23 chromosomes, not the normal 46. But beyond meiosis, when sperm are being produced for spermatogenesis, a couple other things happen to you. Why are they so tiny? Basically, as these sperm are being formed in the seminiferous tubules, most of the cytoplasm is shed. That's what makes them small. These are basically a nucleus on a stick. I mean, you got the nucleus here, takes up most of the head. A few mitochondria there and a flagellum, that's it. As these things are maturing, they shed all the cytoplasm. So there's not much in there. Now, sperm, we know, have to move. Eventually, when they exit the male's body, in order to have successful reproduction, the sperm have to swim. So basically from the end of the vaginal canal, through the cervix, through the body uterus, through the uterine tubes, it's a huge distance relative to the sperm size. They have to do that with the flagellum. So the flagella, it's for motion, movement. It's a bunch of microtubules bundled together. But to get movement, you have to have one thing. To move something, do work on something, you have to have an energy source. Sperm cells are so tiny, they don't have any significant stores inside the cytoplasm. In order for these mitochondria to function and make ATP, that's what mitochondria do, they have to have a substrate, something to work on, like carbohydrate. They think that's what the fructose is for. If you don't have any fructose, the sperm is ready to go, it's got mitochondria. It doesn't have anything to make ATP with. If you have no ATP, the flagella doesn't work. So they can't swim. You're sterile. So the fructose may just be a substrate. It's added to the sperm, packed into them as they're moving past this point. So the seminal vesicles squeeze out the secretion of a lot of fructose. The sperm can use that as a substrate or an energy source. Mitochondria convert it. There you go, you get your ATP, which the flagellum can use, and now the sucker can swim. All right at the junction then of the duct from the seminal vesicles and the vas deferens, they join. And now this new tube, after they connect, is given a different name. This was on your lab sheet. That's the ejaculatory duct. So just change its name. When you pass the vas deferens by the ducts of the seminal vesicles where they join, we change its name now. It's not vas deferens anymore, it's ejaculatory duct. And you'd have two of them. There's again one coming from each side. And I'll mention at this point, the ducts are running through another accessory reproductive gland. This is the prostate. Now you only have one prostate. You have two seminal vesicles, but everything goes through the prostate. There's just one. And it's prostate, one R, not prostrate. That's different. All right, so as you move this fluid through the prostate gland, it's going to dump in its secretions. Secretions from the prostate gland, they account for about 30% of semen volume. Not what seminal vesicles do, but it's still significant. And again, secretions from the prostate gland, it's an aqueous solution, it's a mixture. So it, again, ions and other electrolytes, It's got some prostaglandins in there, which are signals that might aid contraction of smooth muscle down the road. But one of the interesting aspects of prostatic secretions very basic, and a high pH. Normal body fluid is like 7 point something, 7.4, but this is quite a bit more basic than that. Why would you have basic secretion from the prostate gland? Well, it's because of the female reproductive tract. As we'll see, the female reproductive tract, if you go through the vaginal canal, it'll go right through the uterus, right through the uterine tubes. And if you notice on models, the uterine tubes are just open into the peritoneal cavity. They're just open in the abdominal cavity. That is a direct pathway for potential pathogens to get right inside your abdominal cavity and cause all kinds of havoc. You don't want things moving through your reproductive system into your abdominal cavity. So to prevent sort of opportunistic infections, the female reproductive tract is very acidic. It's got a low pH, somewhere about 5 or 4, somewhere there. That acidic environment is to prevent invasion by various microbes. The problem is that low pH also is damaging to the sperm. So it wouldn't do to be getting rid of your microbes but killing the sperm at the same time, you go extinct. So to protect the sperm, it's possible that this basic secretion from the prostate produces a pathway where you neutralize the pH, the acidic pH in the female reproductive tract, and that's protected for the sperm. So the basic pH of the secretion from the prostate help to neutralize the acidic environment in the female reproductive tract so the sperm aren't dead. A couple other things about the prostate. Of course, these ejaculatory ducts will then enter the urethra. Here's the urinary bladder here. So the urethra, as I mentioned in lab, it goes from the bladder all the way out to the penis, but they separate it into different areas. And it's kind of self-explanatory. Where the urethra is running through the prostate, just called the prostatic urethra. But one of the common problems with prostate in males, particularly when they get older, is you get hypertrophy of the prostate. It starts getting too big. You get like occurs in over 70% of males that get older. And also prostate cancers are fairly common. It's the third most common cancer in males. Now, of course, you see why this causes a problem. Here's your urethra. If this prostate starts getting bigger, what happens? Even if it's not cancerous, it squeezes the urethra, makes urination difficult, that sort of thing. So it is a common problem spot in males, and they have various ways to treat it. I mean, one of them, if it's cancerous, they'll put in radioactive beads, which will destroy some of the cells, reduce the size, and they check it frequently. Prostate cancer secretes some chemicals, I can't remember the name off the top of my head, but they'll pick that up in blood tests, get a number of false positives, but it's a very easy way to detect it. Also, when you get older, you get monthly prostate exams. Of course, this thing's about the size of a walnut, that's how they describe it. Here's your anus rectum. So for a second, just go big there. Yeah, it feels about the right size. It's very lovely. But problem area in males. So we take these secretions through the ejaculatory ducts. They'll enter the prostatic urethra, which will then exit the abdominal cavity right here. This short bit right here, that's the membranous urethra we talked about last. It's just really short. This little layer, it's kind of muscle right there, connective tissue. That is referred to as a urogenital diaphragm. It's basically the floor of your abdominal cavity. So the urethra penetrates right there. So that little short bit, that's what we had as a membranous urethra in labs, visible in some models. And then we'll enter into the last bit of the urethra, which goes through the penis. You can call it penile urethra, that's fine, because it's surrounded by that corpus spongiosum. I think that's listed as spongy urethra on your lab sheet. Spongy urethra, penile urethra, the same thing. But before we leave the body here, there's one more of those accessory reproductive glands. It's a little tiny one. There's also two of these, kind of one on each side. They're just kind of above the base of the penis. These are the bulbourethral glands. And they only account for about 5% of the semen volume. And they're secreted a little differently. More or less, the secretions from the seminal vesicle and the prostate, they're just dumped in with the sperm sort of as it goes by, adds the fluid. the secretions from the bulbous urethral gland are released initially, kind of before the sperm gets there. They do two things. When you get these secretions from the bulbous urethral glands, they're basically a mucousy secretion. They do two things. One, they act as a lubricant during coitus, during intercourse. And number two, because in the male, the urethra is part of the two organ systems. It's your reproductive system, but it's also your urinary system. Urine is nasty stuff, right? It can have real high pH, real low pH. It's got all nasty waste material in it. There can be residual urine sitting in your urethra here. If you dump your sperm right in with that urine, the urine can damage the sperm. It's not good stuff to swim around in, I guess. So part of what the secretions from the bulbous urethral gland do, they will flush out and neutralize any residual urine kind of right before the sperm gets there. So it's kind of cleans it out, then the sperm comes through, and there's no residual urine there to do any damage. Then, of course, once we leave the urethra, we're done. That's the end of the male reproductive system. A couple other bits and pieces. Picture that will do. All right. The of course, external genitalia on males, basically the scrotum and the penis. I just want to mention a couple things about the erectile tissue in the penis. We mentioned it last and look at where it was. Look at the cross section of the penis, but I didn't do much of anything else. All right. So look at the little cross section here. The penis there. And just like in line, you've got two columns of corpus cavernosum. I'll draw them up here. I'll do this way. Let's do this. All right, that looks like a pie chart with 3 bits to it. What you have up here, dorsally, there's 2 columns of the corpus, which just means body, cavernosum. And down here, you'll have the urethra around it. You'll have a little layer of corpus spongiosum. And this opening is the urethra. All right, I mentioned in lab, the erectile tissue, what it is. Of course, in the penis, this red represents just fibrous, dense connective tissue all around the outside of the penis, making a kind of septum between the 2 columns of cavernosum. But inside the erectile tissue itself, it's just dense, fibrous connective tissue, very strong, and it has all kinds of blood sinuses in it. So, when you look at it in the microscope, you might have seen, big open areas in here. Those would be, if it were intact, connected to the circulatory system. And of course, the purpose of the erectile tissue, again, is the penis has to function as a penetrating, a copulatory structure when you're using it. But when you're not using it, you don't want it stiff and getting smashed into everything. So you want to get all flaccid so you can cut it out of the way. The erectile tissue is basically working like a series of a bunch of water balloons. Again, if you pump blood under pressure in these spaces, it fills them up, it becomes turgid, rigid. The penis can work as a penetrating structure. If you drain the blood out, it gets all flaccid. You can cut it out of the way. The way it works is kind of cool, sort of in general. You can imagine there's a vein. I don't know if it's on this picture very well. Okay, we got an artery here, and then a little vein up here. So imagine, here's your vein. It's pretty, I'm just going to draw an artery here. It's pretty superficial, and here's your artery. So here's kind of the way it works. If you're getting aroused, you dilate your arteries a little bit that go to the penis. More blood goes to them, and these arteries connect all these blood spaces. So as you increase the blood pressure and blood flow, they'll fill up, and this tissue will start to expand. Well, as the erectile tissue expands, kind of look at the location of the vein here. As the erectile tissue of the penis expands, it's going to take that vein and basically it gets squashed. It kind of gets flattened against the tough connective tissue sheath on the outside. So what you have is an increased inflow of blood, but you're restricting the outflow, so that it increases the pressure in the erectile tissue. and it becomes rigid and cord. Then, when you're done using it, you can just vasoconstrict these arteries. That is, you make them smaller, and it'll constrict the blood flow going in a little bit, and you'll increase the blood flow going out as it sort of deflates, and it becomes flaccid. Now, what's interesting with the control mechanism, we won't look at circulation for a bit in lecture. It's basically exam 2. But I'll just mention now, most of your blood vessels, they have smooth muscle in them, and they can constrict or relax. They call it vasoconstriction, vasodilation. Basically, when you constrict a blood vessel, less blood flows through it, but it increases the blood pressure upstream. So you can control blood flow through these tubes by changing their diameter. So you make it smaller, the term is usually vasoconstriction, because the smooth muscle constricts and makes it smaller. Dilation, it'll get bigger. You have this relaxation constriction in most of the arteries in your body, but most of them are controlled by your sympathetic nervous system. That is, almost every blood vessel in your body is connected to the sympathetic nervous system. And so depending on what the sympathetic nervous system is doing, that'll make blood vessels either dilate or constrict, depending on where they are. The exception, one of the exceptions, is the blood vessels going to the erectile tissue. They're not controlled by the sympathetic nervous system, so it's a little different. These blood vessels are controlled by the parasympathetic nervous system. And again, part of the reason for that is, if your fight or flight response kicks in and you have to start running away or fighting something, you don't all of a sudden, get a big erection and then, that's just not the time because it can get damaged, cut off, broken, whatever. So the blood flow to the erectile tissue is controlled a little differently from all of your other blood vessels. It's controlled by the parasympathetic nervous system. Rest, relaxation time, that's when it can increase, diameter, change the characteristics of the arteries supplying the erectile tissue. That's all we need to do with the male. So we get a little time. I'll start with the female a bit. We won't finish it. We'll get some of it done. It's a pretty short section, so I will start. This won't work. All right, so I'll treat the female reproductive system kind of like we did in the male. Kind of start at the site of gamete production and move through the ducts. So what we'll do, I'll start this today. They've got us finished there. It doesn't take much in the anatomy. Then the only other things we have to do, I look at the development of male and female reproductive systems. It's kind of interesting. The way they can get either male structures forming or female structures forming is, I think it's interesting. It's not genetic, really, as you would think. It's hormonal. That's what causes a penis to grow versus a clitoris and that sort of thing. So we'll look at development of the male versus female reproductive system. And then the only other thing we'll look at is the hormonal or endocrine control of reproduction. That's real simple in a male, but that's complicated in a female because it gets cyclical after sexual maturity. And that'll be it for reproduction. So we start with the female. We will start with the primary reproductive organ, which in the female is the ovary. This produces the female gametes, the oocytes or eggs. And then all those secondary reproductive structures, all the ducts, uterine tubes, uterus, so forth. The secondary reproductive structures, their main function is basically to retain, protect, and allow the development of an embryo and a fetus. And that's what the rest of these structures are for. So you got the ovaries produce the oocytes, everything else is going to protect, retain, and allow development of the embryo and fetus until delivery. Now, weird thing, if you look at this a bit, you'll notice. Again, we're going to start at the ovary. This is what produces the oocytes at ovulation. That's what they're showing you here. Remember, we went through that primordial follicle, primary, secondary, mature. Once that mature follicle gets really big, say right here, there's going to be a release of FSH and LH, and it makes the follicle rupture. And here comes your oocyte. So that is an egg or oocyte. But a couple things to note. In a male, the sperm has no option. It's released at the seminiferous tubule. It's going to go through those ducts, come out the penile urethra. There's no option. But in the female, here's the beginning of the ductwork. And you'll notice they're not connected. Uterine tube starts here, and it's just kind of hanging out over here. Ovaries way over here. There's no physical connection. That's what I said. This, what's kind of white on the board, this is serous fluid. This is just hanging out in that space between the visceral and the private peritoneum. It's filled with fluid that's in your body cavity. So again, the reason it's acidified. Vaginal canal, so outside environment is here. You've got a direct path right into the inside of the body cavity. It's not a closed tube like it is in the male. Now, typically what happens is that there's going to be a pregnancy. Sperm are released about here. They'll swim through the main body of the uterus, through the uterine tube, and typically fertilization occurs in about the first third of the uterus. The egg would be about here somewhere. That's where fertilization normally occurs. But there can be some weird things happening because of the setup. Now, it's very, very rare, but occasionally, let's say this happens. Here comes a sperm, ovulation's a little late, and you get some sperm out here, and they fertilize this oocyte a little early. But then it's possible, and this has happened, this fertilized oocyte, this zygote now, it can miss. So now you have this fertilized egg that didn't go into the uterine tube. That's a big of a problem. Because this fertilized egg, it's going to implant and start to develop anywhere there's a good blood supply. Typically that's the intestines. This, usually it's termed ectopic pregnancy. The term ectopic just means in the wrong place. But you can have ectopic pregnancies that occur somewhere in the abdominal cavity, not in uterus, which is just weird. Now, normally, the intestines, because they're important for diffusion, they have a really good blood supply, high capillary density. A lot of times the embryo will implant there. It will start to form a placenta on the intestine. Normally, those pregnancies can't go to term because most of your internal organs can't accommodate that growing embryo fetus and crush your intestines, that sort of thing. But I think there have been a couple examples where there's some big sheets of tissue in the abdominal cavity called mesenteries. They're actually the membranes holding the intestines sort of to the back of your abdominal cavity. Those are also well vascularized. There have been a few pregnancies where they implanted on the mesenteries. They don't go to term, which is just kind of crazy, I think. Of course, there's not going to be a vaginal delivery. It's got no way to escape, so they have to be cesarean, but it can happen. It's very rare, but because they're not connected, they don't have to go, where they're supposed to. But that's real unusual. Now, looking at this diagram, you say, well, that's happening all the time, man. Any oocyte coming off here, that's going to miss, that's going to be it looks like, it's a little misleading. It looks like you got the opening uterine tube over here, the ovary's way over here. It's like, God, get that little hole. It's not that bad. What usually happens when ovulation approaches, as we'll see in a bit, there are significant hormonal changes. Estrogen, progesterone levels go way up, and that affects the uterus and uterine tubes as well. So what normally happens is the uterine tube where it's flared out right here. What do they call it? Flared out bit? Infundibulum, that goes through your left arm. So it's like a big funnel. And it's got these little finger-like frills or projections sticking out. What were those called? Starts with an F. Fimbriae. That just means finger-like projections. All right, so here's what normally happens. As ovulation approaches, the ends of this uterine tube, the infundibulum, it actually expands. They get engorged with blood, so they get huge. So it's not sitting off here like that. I can't draw it, but it really looks something like this. I think it's more like this. So it's very difficult for the oocyte to miss. It's not like shooting a basket from from your arms or something. So the whole end of the uterine tube does engorge with blood, get much bigger. In addition, there's smooth muscle in here. So these fimbriae, little fringes at the edge, they're kind of like, I don't know, you were little, did you ever play with a toy in a pond or something, and your little bubble starts to drift, you can't reach it, so you take your hand and you do this to bring it back in. That's kind of what the fimbriae do. They start to move. So and this is all fluid, it's not air here, this is liquid. So these things start to move. They do create some gentle currents, which tend to direct any ovulated oocytes into the uterine tubes. So the end of the uterus, it gets bigger, the fimbriae become active at ovulation, again, because of the hormonal changes, create some currents that draw them in. And also the inside of the uterine tube, not shown on here, but have ciliary tracts. Those also create currents. So there's a number of factors that typically, when an egg is released, if ovulation occurs, it's going to make it in the uterine tubes. When it misses, it's very rare. Again, that's because the end of the uterine tube gets bigger, the fimbriae get active, the ciliary tracts are active, and all that creates currents that normally ensure that that oocyte is going to make it into the uterine tube. Now, as we'll see, once an oocyte enters the uterine tube, it's going to move pretty slowly. As we'll see when we develop, it takes like 3 or 4 days for the oocyte to go from here to here. It's a pretty slow movement. But it's going to make its way through the uterine tube, you got one on the other side. We'll just go. And you'll make it to the main body of the uterus. Now, just because we have a lab, there's some different sections of the uterus for lab. I won't talk about it much in lecture, but this part here that's kind of sticking above where the 2 uterine tubes come in, that on the lab was your fundus. This end where the uterus communicates with the vaginal canal, cervix, and the rest of it is just typically called the main body of the uterus. So we'll tell you about fundus. You'll see fundus a lot. The stomach has a fundus. A lot of things have a fundus. Real way you want to find the fundus, and look, they're all going to be hollow organs. You find the outlet to the organ, which here for the uterus, this is where things come out, right? That's the outlet in the stomach, where it goes to the small intestine. You move to the farthest point away from the outlet, that's always the fundus. It's different, you know, I'll just say it's above where the uterine tube's coming. But really, you find the outlet, move as far away as you can, that's the fundus. So again, it's a term you'll see in lots of different organs. So typically, if you have a fertilized oocyte, it needs to make it to the main body of the uterus. This is the only part of the system that can accommodate the growth of that embryo and fetus. You know, occasionally you'll get embryos implanted in uterine tubes. It's a type of ectopic pregnancy. They call them tubal pregnancies. They usually have to be terminated. This uterine tube is tiny. It can't accommodate the growth that that embryo and fetus is going to have. So you're supposed to make it to the main body of the uterus. And this is where the embryo will implant, you know, placenta will form, and you'll carry it for 9 months. But the last thing we'll do today, let's look at the layers of the uterus, because there's a couple interesting aspects of the layers of the uterus. So we start at the outside, kind of just this layer of white. That is just a layer of basically epithelium and basal membrane. That is the visceral peritoneum. It's also called the perimeter. It's just a thin outermost layer covering the uterus. It's, you know, totally like a cell layer thing. Doesn't do much, it's just there. But what I want to look at is the next layer. Most of the uterus, most of its volume, is smooth muscle. We call it myometer. Now here's my question for you. Last semester, you looked at muscle types probably. You looked at cardiac muscle a little bit in histology. You looked at smooth muscle, skeletal muscle, yes? Do you remember the differences? What's the difference between smooth muscle and skeletal muscle? Let's list a couple differences. Is what? Okay, skeletal muscle is striated, so is cardiac muscle. Smooth muscle, no striations, hence the name smooth. What else is different? What? Yeah, there's different nuclei. What else? Here, tell you what, make your smooth muscle contract right now. Can you do it?
No.
Make it relax.
Oh, one is voluntary, one is
Voluntary and involuntary. Smooth muscle is involuntary. Skeletal muscle is the only one that's voluntary. Ready? Watch this bicep. Here, contract now. It's like magic. We're going to make it relax. There we go. So you can control skeletal muscle. It's under voluntary control. Smooth muscle, you can't make your intestines contract on demand, either your uterus. This is where I have a complaint, just personal experience. Why is the muscle of the uterus smooth muscle? Because, you know, baby develops in there about 9 months, and then it's like 11 at night, and your wife goes, honey, I think the baby's coming, and you get in the car, and you drive at 8 miles an hour to Louisville, I usually drive 40, so that's traumatic for me. And then you get there, it's like a 6-hour wait, no, maybe not yet, oh wait, get another hour, it's a pain in the butt. Why don't we have a uterus made of skeletal muscle? That would be way better. Think about it, skeletal muscle is voluntary, right? So you let baby grow, and then your wife goes, honey, I'm thinking Tuesday at 2 o'clock, I'm gonna go ahead and have the baby. And you know, you leisurely stroll up to Louisville, maybe have some lunch ahead of time, and then you go in and the doctor says, "Woman's at 2, you ready to?" "You ready, doctor?" "Okay, go ahead." Boom, out it comes, done. Oh God, that was so much easier. You have no babies born in, you know, back of cabs and snowstorms. It would be a lot easier. Why isn't the uterus skeletal muscle? Why easy? I think it would be. Why not skeletal muscle for the uterus? Seems like a better system to me. You know, rather than popping out a baby randomly, you know, evolutionarily, there could be predators coming. Oh crap, baby's coming too. This would be terrible. Why not make it skeletal muscle? It's voluntary. It would be way better. We should do some genetic engineering. All right, the reason, you know, here's where I use this to explain an aspect of skeletal muscle contraction. Last semester, we look at skeletal muscle for our model for skeletal muscle contraction. You remember troponin, tropomyosin, the sarcomeres, and all that. All right. We don't look much at smooth muscle, except, you know, what you told me. Well, it's not striated, it's involuntary, you know, mononucleated, and that's about as far as we go. But we're going to do an experiment here. What we're going to do is have this junk upstairs. This is a muscle transducer. Doesn't quite look like this. It's just a machine with a hook coming out of it. If you pull on this hook, it sends a signal to the computer, and you get a line drawn that is relative to the force applied to this machine. So it just converts mechanical force into electric signal you create. It's got a little threaded part here, so you can raise and lower it, and it's just sitting on your bench. This is just a muscle apparatus here. It's just basically a upside down test tube, a test tube stuck on a table, and it's got a little hook here. Now, as a common muscle prep, what they'll do is they'll take a piece of muscle, they'll stick it here. It's attached to this hook, and you fill this thing with, normal saline, so the muscle is all fine and happy. So when you stimulate this muscle, it's going to pull down on the hook, and you can measure the force that it develops. Now, what we're going to do is we're going to take a piece of, just a piece of skeletal muscle, imagine something like a bicep. So we take that muscle, we attach it to these hooks, just as loose as it can be. I mean, it's just barely, hung between those two hooks. Then we'll take a little stimulator and a couple of electrodes. You could use a battery, a little crude, but it'd work. Send a little electrical signal through there, the muscle will contract. So all we're going to do is we'll take this muscle, and what I'm going to do is take a little ruler, and I'm going to take a ruler right here. So I'm going to measure how long this muscle is, and then I'm going to stimulate it, and it will contract it. And what that will produce is a length tension curve. So on the x-axis here, it's the length of the muscle. Physically, before you contract it, measure it with a ruler. This long, this long, this long. So we start with a real short muscle. This end is short. This end is just relatively speaking longer. The y-axis is force, is tension. So we take our short muscle, it's not stretched at all between the hooks, it's just barely sitting there. We stimulate it, it contracts, we get a little bit of tension. Then what we're going to do, because this is threaded, we can kind of crank this little threaded bit up, and we'll raise this whole half of the apparatus. So it's now up here somewhere. And the hook's up a little higher. So we're actually going to take that muscle and stretch it up a little bit. So the muscle is now literally stronger. We're not stimulating, we just took the muscle and pull it a little bit. We stretch it out a little bit, stimulate it again. See how much tension it develops. We keep doing that. We crank this thing up some more, raise the hook some more. Stretch the muscle out more. Stimulate it again. And what we'll do, we keep doing that, make the muscle longer. Every time we stretch it, we stimulate it, contract. Stretch it, stimulate it, contract. Stretch it, stimulate it, contract. And we plot the force of that contraction. And then just connect the dots. You're going to get a curve looks something like this. This is sort of a standard length tension curve. What it means, in the middle here, that's maximum tension. Over here, there's basically no tension. Over here, there is no tension, zero tension. You stimulated it, didn't do anything. This is what skeletal muscle will do. Now, the reason why, you have to think back to that contractile mechanism of skeletal muscle. You remember you had the sarcomeres, you had the little Z-line on either side, the little actin filaments, and then the myosin filaments in between. All right, let me look what they look like at the extreme, because it's all about the sarcomeres. At this end here, when we've really stretched the muscle out as far as we possibly can, here's a Z-liner disc, here's a Z-liner Z disc. This represents the actin filaments. And I'll use red to represent the myosin filaments, kind of held between. So what we've literally done, at this is your sarcomere, we've stretched. Right now there is no overlap between the myosin filament and the actin filament. If there's no overlap, can you get into those cross bridges to form? No. So if you stretch a skeletal muscle out to this point and you stimulate it, is it going to contract? No, there's no tension. If they can't interact, nothing happens. That's what you got here. Then if you look at this end, all the way to the left, where there's no tension here, we let that thing get as short as it can, and now you're going to have something like this. Here's your Z disc, here's your Z disc, here's the actin filament. Here's your myosin filaments. Basically what happens here, as those filaments come closer and closer together, eventually the myosin filaments crash into the Z disc. It is a protein disc, and they crumple, crumple, crumple. The actin filaments come together, they go boom, crumple, crumple, crumple. The technical term is protein interference. But yeah, the fibers literally go crunch. And eventually they crunch up enough, doesn't matter if you stimulate it, it's not going to contract anymore. That's over here. So if you look right smack dab in the middle, what you have in that middle example, you've got the maximum amount of overlap between the actin and myosin, so you can make the most cross bridges, but the proteins haven't started to crumple into each other or interfere with each other yet. That's max tension. This is typical of all striated muscle. Skeletal muscle will do this. If you overstretch it, little tension. You get it too short, little tension. They're usually set up the way they are in your skeletal system, so they're kind of at this point when they're contracted. But they're sensitive to stretch. They have a length tension curve. We'll see this with the heart too. It's kind of important with the heart. But what about skeletal muscle? What about smooth muscle? Does it have sarcomeres? No, remember that's why it's smooth. Sarcomeres are just that repeating pattern of protein that give it a striped appearance, on a micrograph, on a histology slide. Smooth muscle, it still has actin and myosin and so forth, but they just randomly spread around. There's no pattern to it. So if we do the same thing with smooth muscle, so I come in here, reset my apparatus, take that piece of skeletal muscle off, do exactly the same thing, but now I'm going to put in a piece of smooth muscle from a uterus, let's say. Oops. And I do the same thing. Let it get really short, stimulate, stretch it, stimulate, stretch it, stimulate, stretch it, stimulate. Do the same thing, plot the same points, I get something that looks like this. It doesn't change. This is why you use smooth muscle in most of your hollow internal organs. If you stretch out smooth muscle and stimulate it, it still generates the same amount of tension. So it's insensitive to stretch, which is why it's found in uterus. Because think about it, if you made your uterus out of skeletal muscle, because you're doing some obscene, genetic engineering, everything is seen fine. The uterus starts off this big, and then the baby develops, it stretches, it stretches, it stretches. 9 months later, you send a signal to the skeletal muscle in the uterus, and what would happen? Nothing, and you'd be stuck pregnant the rest of your life. It would be really uncomfortable. It wouldn't work. If you take skeletal muscle and you stretch it that much, you can't do it. It would overstretch. It wouldn't have any tension. There'd be no delivery. That's typically why, again, intestines, your urinary bladder, your stomach, your uterus, it's smooth muscle. It isn't sensitive to stretch. You can stretch it like crazy, and it doesn't matter. When you stimulate it, it's still going to contract with about the same amount of force. Very insensitive to stretch. That's why it's in your hollow internal organs. That's why they usually call it visceral smooth muscle. It's associated with your viscera, your guts, your smooth muscle. Your internal organs. All right, so much for my convenient labor experiment. It wouldn't work. All right, so myometrium is certainly the bulk of the uterus. And again, it's pretty small outside of a fist or a pear where it starts, but Junior's going to implant in there and it's going to stretch it out tremendously. But 9 months later, you send a signal to it, it's still going to contract with sufficient force. The innermost layer we won't do anything with today. We will look at this one quite a bit when we look at endocrine control. The inner layer is the endometrium. The endometrium is the layer of the uterus that is very sensitive to the sex hormones estrogen and progesterone, and it's the endometrium that will actually eventually form the mother's half of the placenta, but it changes depending on the hormonal state. Endometrium will be really thin, or it can get really, really thick just before implantation and pregnancy might occur. I'll go through some of the endometrium, I guess, next time. So all we have left to do with the female on Tuesday, we'll basically go through vaginal canal and the external genitalia. Once we're done with that, we'll do a comparison of the development of male and female with what they call symptomologic structures. And then we'll look at endocrine control of male and female reproductive systems. So we will have reproductive systems wrapped up on Tuesday, it looks like. Questions, comments, send an email if you need to. start development next week. We only go at, so we should be done with development by next week. So that's probably what your quiz or exam will cover. Endocrine, male female reproductive and development. Okay, those are all in. About half the exam will be endocrine, so we spent the most time. Have a fun and exciting weekend. Send me an email if I forgot to do something. This video for huge.